section, we're going to talk about the cell wall of prokaryotes, specifically bacteria. We're going to talk about what they're composed of, the characteristics of gram-positive and gram-negative cell walls, and why the gram stains the way that it does. We're going to talk about atypical cell walls, specifically those of mycoplasma, archaea, and acid-fast bacteria. Then we're going to end up with damage to the cell wall, since this is what an awful lot of antibiotics and disinfectants, sterilants do, is damage the cell wall. For this discussion, we're going to be referring heavily to stuff that you learned in Chapter 2. You're probably thinking, oh no, Chapter 2! Didn't I warn you that it was going to come back? It's not too bad. In any case, the main component of bacterial cell walls is peptidoglycan. Okay? So the glycan comes from sugar, peptido from protein, so it's a sugar-protein composite. Now this diagram here is the sugar portion. We have modified glucoses, maybe it's glucose. We have modified glucoses that are hooked together, and they compose the sugar part of the peptidoglycan. Now one of the units, modified units, is NAG. I'm not going to make you remember the N-acetylglycosamine, but here is one part that's been modified, and we also have an amine group with some other fun stuff on it. Then NAM over here has this part modified from this part, and it also has this amine group. And we get this repeating, NAG, NAM, NAG, NAM, NAG, NAM. Now, in the book, they represent this as kind of a water noodle with brown and pink segments. Okay, so that's the sugar portion. These little balls are amino acids that are hooked together to form peptides, a tetrapeptide and a hexapeptide that hook the water noodles together. You put these all together and you get a cell wall made out of peptidoglycan. Now with this one, this is a model of a gram-positive cell wall. It's got a very thick layer of peptidoglycan. Okay. Now, if you look at a gram-negative cell wall, it's got a very narrow band of peptidoglycan, and it's got a membrane on the outside. Okay. So what happens during the gram stain? With a gram-positive cell, the stain gets into the thick cell wall. Then when the alcohol comes across, it doesn't really do too much to it unless you leave it on for too long. The cell wall can't stand it for inevitably. Okay. So it retains the first stain. That's why it keeps the crystal violet. Now down here with our gram-negatives, what happens when the stain comes in, okay, stains this outer membrane, it's got phospholipid heads, so the positive stain is attracted to the negative heads of the outer membrane. Then when that alcohol comes along, that alcohol busts up that outer membrane, washes it clear away. Then you put the secondary stain on to stain the cell. That's why they stain differently. Now there's also differences that I want you to remember. Gram-positives have a molecule called tachoric acid. That's part of how biochemically we recognize gram-positive cells. Gram-negative cells have a molecule embedded in that outer membrane that's called LPS, lipopolysaccharide A. Actually, it's lipopolysaccharide. Lipid A is the lipid part of that. Now, why do I bring that up? In your body, if you get a lot of LPS floating around in your bloodstream, your immune system goes nuts. It sees it as a vast amount of invading bacteria, and it overreacts, and you go into what's called septic shock. So if you have a gram-negative infection, we have a tendency to want to be very careful about how fast we kill those bacteria so that we can prevent septic shock. So in review, you want to remember that gram-positives have a thick peptidoglycan cell wall that has tachoric acid in it. Gram-negatives have a thin peptidoglycan cell wall with an outer membrane on the outside in which there is LPS. Now on to atypical prokaryotic cell walls. Let's start with mycoplasma. Mycoplasma are bacteria that have lost the ability to make cell walls. So when you look at them under the microscope, they form these almost amoeba-like blobs. They have a tendency to still be kind of long and skinny, but they don't have cell walls, and they can wiggle through filters. This is something to remember later on when we talk about control of bacteria. It also makes it so that they are resistant to antibiotics that attack cell walls. Makes sense. They don't have cell walls. Now let's talk about archaea. Archaea have a type of cell wall component called pseudopeptidoglycan. It's very similar to peptidoglycan, but instead of NAG and NAM, they have NAG and NAT. And over here, I've got a lovely picture of what NAT looks like. That's all you're going to have to remember. Now, for acid-fast bacteria, of which tuberculosis is caused by an acid-fast bacterium, so is leprosy, they form a waxy coat around their cell wall, and it's made up of a lipid called mycolic acid. Remember that their fatty acids can have an acid group on the end of them. Well, this mycolic acid performs approximately the same function as a capsule. It prevents phagocytosis. It also means that acid-fast bacteria are difficult to stain. That waxy coat does not have a real strong negative charge, therefore your basic stains don't want to stick to it. So we have a different staining protocol for staining acid-fast bacteria. Mainly the acid-fast staining protocol is only used in TB clinics or other places where you're expecting to see acid-fast bacteria. Generally you do a gram stain first. It's easier, it's fast, breaks your bacteria into two main groups. Also, because of this long lipid chain, it is expensive for the bacterium to make. So it slows down the growth of your acid-fast bacteria. That's why tuberculosis is such a long-term chronic disease. It resists your immune system and it grows slowly. Let's talk about how you can damage prokaryotic cell walls, specifically bacteria. Lysozyme is an enzyme that is naturally found in human tears, saliva, etc., that breaks apart the NAG and NAB subunits of peptidoglycan. This causes holes in the cell wall. This also leads to the contents of the cells leaking out. Now, some antibiotics go about it a different way. They attack the tetrapeptides that hold the peptidoglycan, the glycan units together. Penicillin is an example of this. Alright, that's it for part 1, and we will come back and discuss the rest of the prokaryotic cell in part 2.